Group name. Enkel.
Yeah. We have a group name.
You do have a weird. That's the group name. That's what I'm looking for. Oh, there you go. Okay, we're going to talk about this. So, this is actually really dead simple. It really is. Okay? So, we're going to go through this together. So, we're going to draw this. I'm recording this. We're going to draw this. So this is the general overall pathway that happen with both T cells and B cells. We start with this common lymphoid progenitor. Technically, the hematopoietic stem cell is here. And so because hematopoietic stem cells can become anything, this is why we call this a common lymphoid progenitor, because these now can only become lymph cells, lymphocytes, right? B cells and T cells. Does that make sense? Anna?
Yeah,
Ann?
so this is, you're saying the common lymphoid progenitor is a hematopoietic stem cell?
So what I'm saying is this is the hematopoietic stem cell right here. This can become mast cells, this can become PMNs, this can become lymphocytes. But if it's going to become lymphocytes, it then is called the common lymphoid progenitor, because as they go through the maturation process, as they differentiate, they're going to start changing their DNA so that a mast cell is going to be a mast cell, and a dendritic cell is going to be a dendritic cell, or a monocyte, or something like that. So now this common lymphoid progenitor is only going to be lymphocytes, B cells and T cells. Okay? So we have this common lymphoid progenitor here. And the reason this is important is because we're going to talk about what happens when errors happen in later chapters, okay? So here we have a common lymphoid progenitor. They're going to start differentiating when they go to the bone marrow. They're going to start going down the path to become bone marrow, a B cell. And when they migrate to the thymus, they're going to start going down the path to start migrating into the T cells. So we're going to start this, and then we're always going to rearrange the most complicated chain first. Okay? For B cells, it will always be the heavy chain. For T cells, it will be the beta chain. Because the B cell heavy beta chain and the B cell heavy chain and the T cell beta chain look almost exactly alike except for the constant region areas. For B cells, we have multiple constant regions. For T cells, all that constant region is going to do is hold that variable region up for display. So we're going to have a common progenitor, and then we're going to test it to see if it can send in a function. Okay? So once we test it and check to see if it's functional, because remember, we're taking rando, B, D, and J regions, snapping them together, adding extra nucleotides in between each set, it may not work. It may look like a B cell receptor, but might not be able to function. Or maybe the different signaling molecules didn't associate with it correctly. All of that can happen, okay? And if that doesn't work, then what do we do with it? We're going to kill it because it's not functional. We have essentially two chances because you have two chromosomes. So if the first rearrangement doesn't work, they'll rearrange the second one. If the first rearrangement works, we call it allelic exclusion because we only want one heavy chain expressed or one beta chain expressed. Because remember, every B cell and every T cell only makes one receptor. So if it can't do this, it dies, okay? So then, yeah, Mary?
So each pre-B cell and pre-T cell will make one heavy chain, and if that doesn't work, they get a second chain.
One heavy chain or one beta chain.
Okay.
And if it doesn't send a signal and they can't function, they do get a second chance. If the first rearrangement works, then they don't rearrange the other one, and that's what allelic exclusion is. Does that make sense?
Yeah.
Okay, so the other thing is, the way that I remember this is because I'm dyslexic and I don't remember stuff, okay? Is I remember a pro has a 0 on it and nothing on the O. This is nothing here. This has just gone down from a common lymphoid progenitor to either become a T cell or a B cell. It's either going to stay in the bone marrow or it's going to go to the thymus. Okay? Pro doesn't have squat on it, 0. Okay? Then we start differentiating the pro into a pre. Okay? And that's the only way I remember. So I find little mnemonic ways of remembering stuff. Okay? The other thing that's important and that the book talks about is that it requires IL-7 for this to happen. IL-7 is going to be helping these things differentiate. Okay? So, we have the common lymphoid progenitor, which is differentiated from the hematopoietic stem cell. So now we have this common lymphoid progenitor that now will either be a B cell or T cell. Depending on where it goes as a pro cell, will make it differentiate into a pro T cell or a pro B cell. Once it gets to where it's going to start activating its RAG1 and 2, the recombinase activation genes, RAG, RAG, recombinase activation genes, 1 and 2, and they come together to form the recombination enzyme that allows us to actually cut and delete chromosomes. This is the only place in the body where we're going to delete sections of the chromosome. And if we checked on a gel, we would see that the germ line would be that long for the chromosome, and once it rearranges for a B cell, it would be that long because we've deleted that much DNA. For T cell, the beta chain would be that long because we deleted that much DNA. Because we have literally cut and thrown away pieces of the genetic material. Okay? Does that make sense? So then, in order to get this first chain rearranged, because we're going to test the beta chain and the heavy chain, we need, I like to think of it as a fake other receptor. So it's a pseudo whatever, a pseudo light chain or a pseudo alpha chain. It's like putting a donut on a car temporarily. We're going to just throw that on because we know that will work. We know that if we put that pseudo chain up with the rearranged receptor, if the receptor is good, that pseudo chain will allow a signal to be sent. Does that make sense? Okay? So we just are gonna, so we know the pseudo chain will always work. It doesn't work well like a donut. A donut will get you on your car to where you need to go, but you don't want that as your part of your car. You don't want to keep it on your car all the time. It's not functional. So what we're going to do is we're going to test this heavy chain or the T cell receptor, and then once we have that combination, then we have RAG recombinase do what? Rearrange the other chain, right? And then we test those. And this is where T cells and B cells really start differentiating, okay? So, and then once we have a receptor on, then it starts differentiating from there. So let's go through, I like to go through the B cell maturation process first, because to me, that's the easiest way. It's the most straightforward way. So here, we're gonna have the hematopoietic stem cell, okay? And it's going to become a common lymphoid progenitor. You don't need to, I mean, again. So, what is on the surface of the common lymphoid progenitor? Nothing. It is just, it looks like a lymphocyte. So this is the nucleus, okay? So this common lymphoid progenitor is going to stay in the, to be a B cell, bone marrow. Okay, and then it's going to start getting signals to start differentiating. So this is now, because it's in a bone marrow, what kind, it just got into the bone marrow and it's getting signals that it's now a B cell. And because there's nothing on it, It's just being told, because you're in the bone marrow, it's receiving genetic signals that you are now a pro B cell. You are going down the differentiation path of B cells, not a T cell. Anna? Is it Anna or Ann?
Yeah.
Anna, got it.
You said in the last slide that in the second proliferation step, that's what differentiates between B and T cells?
So again, what I want you to know is really it separates at the pro stage. Depending on where these cells go, they're the common lymphoid progenitor. It makes sense if we call this a common lymphoid progenitor, that they can be either B cells or T cells at this stage. Okay? So, because they can be either B cells or T cells at this stage, depending on where they go to mature, is going to determine what separation, what pathway they're going to go down. Okay? So here, we have this common lymphoid progenitor. This is going to stay technically in the bone marrow. And it's going to start receiving signals that you are now a B cell. You are in the Air Force now. You are in the Navy now. Air Force and Navy are different, right? Air Force goes one place and Navy goes another place. T cells go one place, B cells go another place. So now it's going to start saying, we want you to start recombining what? Do we want the beta chain to recombine? No, the heavy chain. So we are going to select the chromosome that has the heavy chain on it. You guys don't have to know the chromosome names where the heavy chain, the beta chain, the alpha chain, the light chains are. You don't have to know those chromosomal names, okay? Just know that there's differences where those chromosomes are. And in this case, we're going to go to the heavy chain chromosome, and we're going to start differentiating this into a pre-B cell. So we're going to differentiate this into a pre-B cell, and a pre-B cell is going to activate what enzymes? RAG. So this is where RAG 1 and 2 get activated to start generating the heavy chain. Now, the constant regions are just constant, okay? What we need to do is we need to make VDJ recombination, and that's where VDJ recombination is occurring. Because RAG 1 and 2 are being activated, we're creating the heavy chain. So we're taking a random D and a J and snapping them together, and then we're taking a random V and snapping it to the DJ, and now we have an exon, okay, that is the variable region. And we simply transcribe the VDJ exon to the first constant region, that is, we call this downstream. There's more isoforms of each antibody, but the 5 classes are mu for IgM, delta for IgD, gamma for IgG, epsilon for, you guys pick up really well, and alpha for, okay. So all we're going to do is we're going to transcribe this exon and the mu constant region. We're going to snap this together into one mRNA. To make variable region. Does that make sense? So now we express this on the surface of the cell. This is the variable region. And we put the what chain? We have to, now, do we put a light chain up? Not here. What do we put up? The pseudo chain, the pseudo light chain, because we're just testing this to see if we put it together correctly. Okay, and also to see this can't send a signal, we're going to have to make Ig alpha and Ig beta so that if it binds to something, we can send a signal. So we're going to put a mu heavy chain up here. Let me see if I can get colors. Oops. We're going to put a pseudo alpha chain, or pseudo light chain. I'm sorry. And I don't know why it does that. Every once in a while when I press too hard, it just makes that line. Pseudo light. So there's no specificity for that light chain. It's just something that will send a signal if that heavy chain has been rearranged appropriately. right? So now we have a pseudo light chain. We're going to test it against self in the bone marrow. And if it can send a signal, what happens? We get survival. So as long as that IgM rearranged can send a signal and bind lightly, and this is where I like the book doesn't say that this is positive selection, but technically I think this is positive selection. If this can send a signal, we positively select this B cell to go on. If it can't send a signal, what do? We could rearrange the other one, see if we could rearrange that. And again, I'm not certain how much that happens. It might just be one rearrangement process. I'll go back and double check. But there are technically two heavy chains. Technically, if this first one doesn't work, we could rearrange the second one, and then we could see if we can get a functional B cell. If neither of those work, we'll kill it. Okay? So, we're going to say that this heavy chain has now successfully been rearranged, can send a signal, a light signal. If it binds too strongly to self, we're also going to delete it. Okay? So, we're going to, this is sending a signal into the B cell that says this is selected. The other thing that I want you to understand is that technically, once it's selected, we're going to actually start clonally expanding this B cell with this light chain, or this heavy chain, the specific heavy chain receptor, because it's hard to make a heavy chain. And because of the way diversity works, every B cell, so we clonally expand this pre-B cell, and now we're going to make this into an immature B cell by rearranging a light chain. But let's look at this. This heavy chain, this is going to, I wish I could, I'm going to have to figure out how to copy and paste these on this. Okay. Okay. We've made identical clones of that heavy chain. And now we're reactivating RAG. Okay? We're reactivating RAG to what? Do what? To make the light chain. Which light chains are rearranged first? The only way I remember this is alphabetically. What? So kappa gets rearranged first. So we actually have 4 chances of making a light chain. So, we're going to reactivate RAG 1 and 2. We're going to, instead of having a VDJ, they're less complicated than the heavy chain or the beta chain. The alpha chain and the light chain only have V and J, no D. So we're going to rearrange that, okay? And we're going to throw up a light chain. Now, each one of these clones of that, we're going to have this light chain generated. Now we have a light chain that looks like this. Okay. Now we're going to have a light chain on this one that looks like this. Now we're going to have a light chain on this one that looks like this. Okay. Now we're going to have a light chain on this one that looks like this. So each one of those, we have a functional heavy chain, and simply by throwing a different light chain on there, we've created diversity already, another level of diversity. Each one of those B cell receptors recognizes a completely different antigen, because the antigen receptor has to be 2 parts. You're going to have 1 part from the variable region of the heavy chain, 1 part from the variable region of the light chain. If you change this light chain to this, or this light chain to this, or this light chain to this, it's gonna change the antigen binding shape that it binds to. Does that make sense? Jessica?
Yeah, so you're saying those are all kappa light chains, and
So,
they're just
each
shifting
one of these goes through its own immature step of creating light chains. It might be that this one made a light chain on the first attempt and that this is a kappa light chain. Okay? This one, however, made 2 kappas that were unsuccessful. Threw away the first kappa attempt, didn't work. Tested it, didn't work. We know the heavy chain works because the heavy chain worked in the pre-step, right? So we know the heavy chain is functional. So if the light chain doesn't work, we know it's a light chain's fault. So the first time attempt, we make the kappa chain, then we rearrange the kappa chain. If it doesn't work, then we rearrange the other kappa chain. Both attempts didn't work. So the next time we're going to make this as a lambda chain. And how do I draw lambda like this? Is that lambda? A lambda chain. So this is the first attempt on a lambda chain and it happens to work. So now we stop. Okay, Savannah?
So for the recombination for the light chains, is it going to be using the same chromosome as it did for the heavy chains?
So we'll show, no, it's a different chromosome for the light chains. It's a different chromosome for the kappa chains. It's a different chromosome for the lambda chains. So the genetic transcription factor, the RAG enzymes know which chromosome to go and where to look to find the genes to rearrange. That's their function, okay? Does that make sense? So, does that make sense, Mary? Again, it's not Mary Grace, it's Mary Gates. Yes. Mary Grace, Mary Gates, dear God. Okay. Okay. Uh-huh. Does that make sense? Okay. Question.
What part of the light chain introduces diversity? Would that be the J segment?
So we're gonna talk about additional diversity in a little bit. So again, when we talk about the alpha chain, and again, this is just for B cells. So this is just for B cells. So when we're making this light chain, so the first kappa attempt worked, we have a functional B cell receptor. This one went through kappa, didn't work. Kappa, the other chromosome didn't work. First lambda, we generated this. This one went through kappa, kappa, lambda, and then the last lambda worked. This one went through kappa, kappa, lambda, lambda, and none of them worked. So what are we going to do? We're going to delete it. Does that make sense? Is this coming together? Are you guys getting this? Yes.
Okay, sorry, I'm still
No, no.
So when
If you're confused, everybody's confused.
So when I read the book, it said that each B cell either had a kappa light chain or a lambda light chain.
Correct.
So I'm getting confused when you say kappa, kappa, lambda, lambda. Like,
So what it
do you mean by
did
that?
was it rearranged the first kappa in the chromosome. The RAG recombinase went to the kappa region, rearranged a kappa, and created a light chain. We tested it against self, didn't send a signal. So we threw that away, and then we tested the other kappa. We rearranged another kappa. Only one kappa, that didn't work. We're going to rearrange a second kappa. Didn't work. Throw that away. Then we're going to go to the lambda chromosome. We're going to rearrange a lambda. Didn't work. Rearrange the other chromosome. Remember, you can hear it, mom and dad. There's a lambda chromosome, a lambda chromosome. We rearranged this one, didn't work. We arranged this one unsuccessfully, didn't work. We have no light chains left to rearrange. There is not a functional B cell here, so we will delete it. But we have 3 functional B cells that did successfully rearrange light chains.
And so you would rearrange kappa first, and then if that doesn't work, you would then try to rearrange lambda, and then if that
You
works,
rearrange kappa, the first chromosomal kappa. All the rest of them are excluded if this one works. If this one doesn't work, what's the next to rearrange?
would you rearrange the second kappa?
Yes,
Okay.
you would rearrange the second kappa.
And you have 2 pairs because one's from mom, one's from dad.
Exactly. 2 chromosomes. If this gets rearranged successfully, we go with this. Allelic exclusion. Only 1 light chain is created, not 2. Courtney? Are you sure? You're not looking like you're good. Okay? Okay.
So, in the drawing you did with the U and all those, is it the same for the VJ, but it's just the kappa and the lambda?
We're going to go over that in just a little bit.
Is it the same, just it's because they're different chains, they're different?
Yes.
So
They
there's-
have fewer, so the light chains and the alpha chains have fewer, they don't have the D regions. So they only have B and J's. Why? Just think of it, I like to just think of it as they're kind of well, T cells is not true. I mean, they're important aspects of the antigen binding site. You have to have the variable region created. And they have CDR region, like very active hypervariable regions. We'll talk about how that's created here. But I want to walk through how this gets created before we go on to the VDJ rearrangements. But we're going to go back over that still yet. Okay, that's still coming. But I just want you to understand that here we have VDJ recombination going on. When we go to the kappa and the lambda light chains, we just have VJ because it's a light chain, okay? And once we create a successful light chain, then we have an immature B cell. A successful immature B cell. Okay? So here we have successful immature and why is it an immature B cell? Karen?
So when you make first the heavy chain and then add on the light chain, you go through both positive and negative selection, right? With that.
Technically, kind of, yes. You can think of it if it binds and sends a signal. So B cells and T cells kind of go through positive and negative selection differently
Yeah.
than they go through positive, and so yes, it's true. If it can send a signal, it's positively selected and it can go on to progress. If it can't send a signal, it's negatively selected. It's different kind of between negative selection is because we're just going to kill it. Well, we'll kill it in positive selection. So we'll just say we're killing it in positive selection because it can't send a signal.
But like, so when you create a heavy chain, are you testing who binds to non-self and then also testing if it doesn't bind to self?
So at this point in time, if it doesn't bind to self, it can't send a signal. It's not going to function. We're going to kill it.
Okay,
Anytime the signal can't be sent, we're just testing the mu chain. We're not testing how, technically for the heavy chain, we're not really testing how high it binds yet because we don't have a fully functional receptor.
Okay.
We're going to test those receptors once we get the light chain made. And as a matter of fact, that's a good point. So now we're going to go through, we've created positive selection. It can send a signal. It can continue to migrate into the path of B cell maturation. The mu chain, the heavy chain, has successfully been rearranged. Checkpoint, they call it a checkpoint in the book. So we passed successfully the first checkpoint, or we positively selected that B cell. Okay? This pre-B cell to go on.
Yeah, so like it's essentially like you make the heavy chain, you do positive selection. If you're successful, you move on to make the light chain. Now you have a complete PCR and you do negative selection.
And you can test, you're going to still do positive selection, can it send a signal?
Yeah.
Because this one couldn't send a signal, so it's deleted in positive selection because it can't send a signal. Now we're going to test these in the bone marrow to see how well they bind. Do they bind to self too strongly? Okay, now this is where, again, a little bit of added complexity, but if you guys think about this, it makes sense. This first one, the first kappa chain, was successfully rearranged and sent a signal. Okay, but the problem is it binds to self too strongly. Okay, it binds to self too strongly. This is a lightning. Okay, so what the B cell does is, let's not waste this resource. We have 3 more chances to downregulate that light chain and rearrange the other kappa chain. Test that. Oh, that wasn't functional. We didn't create another one. Throw that away. Test the next lambda chain. Oh, that worked. Go put that up. It sends a signal and it doesn't bind to self too strongly. So now we call that receptor editing. And in receptor editing, we're going to go in, we're going to remove this light chain. We're going to put up the appropriate, and now, let me get back to here. Okay. Now we're going to put up the first kappa chain bound to self too strongly. Now we're going to put up a second kappa chain, didn't function. Now we're going to put up a third one, and the third one looks like this. Okay, and so now this is the first lambda attempt, but it's functional. So now we have one that binds, sends a signal, and doesn't bind too strongly. Does that make sense? If it's gone through, if it binds to self too strongly, and then it goes through the next kappa, and goes through the next lambda, and none of them function, B cell is destroyed. Does that make sense?
So, how is that different than the one that got X'd off?
So in this case, this never sent a signal at all. There was no signal sent. This was a dead receptor.
So it didn't even try to switch the...
Oh, you're right. It could have. It could
Do
have.
they all go through each of them?
Yes.
And whatever
We're just
sticks
saying that this one never generated a successful receptor ever through all. Yes, and every time there's an unsuccessful, here we can say that we went to the, I think, third rearrangement. We went kappa, kappa, lambda. It would have one more attempt to make a successful receptor.
And
So that was good at least.
if it
You
goes
did.
through all four and it still doesn't send a signal, then that's when it goes.
Again, because we know from the pre-B cell test that the heavy chain is functional, it's the light chain that's at fault. Not me, it's all you. Okay? Are we good? Does that make sense? Yeah?
Okay, so I'm a little confused on like what the kappa and the lambda are. Like, are they recombinations of like B and J segments?
So what they are is just a way of naming light chain regions in a chromosome. So it's just a nomenclature. This is the region for this light chain in this chromosome, and we're going to call that the kappa light chain, okay? And then on another chromosome, we're going to go back and see what chromosomes those are on in just a minute, okay? It's just a different chromosome, and that's where the lambda regions are. And that's where the RAG recombinase can go in and start rearranging those.
So each kappa, like a kappa in my gene is not the same as a kappa in Elise's.
No. The kappa in your, no, that's wrong. The kappa in your genes are exactly like the kappa in everybody's B cells have the same heavy chain in humans. Everybody's B cells have the same constant regions. We have mu, delta, gamma, epsilon, alpha. Everybody has the same. Your antibodies look exactly like Elise's antibodies. That look exactly like Emma's, that look like exactly like Usain's. Everybody. Okay? Everybody. Usain. Sorry. I can take antibodies from you and put them into anybody in this room and give them, we call it passive immunity, because your antibodies will bind to the same antigens that you created them to. You might have been exposed to tetanus. So if somebody is starting to have tetanus symptoms, I can give your antibodies against tetanus to help absorb the toxin that's causing the problem in this person. Now, again, depending on the history of your immune response, you may have never seen, again, the person who's having, so Courtney has never had her, you've never been vaccinated. Okay, so she's never been vaccinated. She stepped on a rusty nail. She started having anaerobic infection. Her organism started producing a toxin, and she started to exhibit symptoms. We don't have time. We don't have 5 days to wait, because she's going to die unless I can do something. You have had all your vaccines, you've been exposed, you have an antibody neutralizing antitoxin titer. I'm going to take them out of you, and they bind to the same epitope that this toxin, because the toxin is the same from the organism. So the antibodies that are neutralizing for you will be neutralizing antibodies for Courtney. So I give them to her, I'm going to absorb those toxins from her system so she doesn't progress wrong. Does that make sense?
Yeah, I just like.
So now the thing is when we say that every antibody will only have a kappa or a lambda chain, that's because once we rearrange one successfully, that is always the light chain that is made. It will be the light chain that is made for that B cell receptor. It will be the light chain that's made for the antibodies that are secreted from that B cell. So when this B cell, once we have a successful rearrangement of these light chains will then be the light chains that are on the B cell receptor and the light chains kappa light chains. So we can actually follow specific clones of antibody responses by looking at the kappa lambda. We can go in and check at the VDJ or the VJ recombination. As a matter of fact, one of my favorite authors died from a leukemia, a lymphoid leukemia, where his B cells started making excessive kappa chains. As a matter of fact, it started just making excessive kappa chains to the point that he started just secreting them out of B cells. And it made his blood so viscous, his heart had to pump sludge every time it pumped because there was so much protein in his bloodstream. Does that make sense? So, normally we associate them with the antibodies. All of these things, every step along the way can have something go wrong, run amok, cause a problem, not be functional. All of this, we're going to learn this in later chapters, okay? Louise.
So in the end, would all of these, like all 4 of these cells, either
So there's
be
only 3.
eliminated or
There's only
have the
3. This
same-
one's dead. This one's dead.
Will they all? Is there only one option that works, or would they all be different? So is it coming from the same heavy chain?
Okay, so is this one different than that one?
Yeah.
It is. It has the same heavy chain, but a different light chain, different antigen binding site. So this is another step in diversity. The first step in diversity is simply recombination. Which V section do we add with which D section do we add with which J section on the heavy chain? Which V section do we add to the J section on the light chain, the alpha chain? So that's the first step in diversity. The second step of diversity is simply what chain do we add to the other chain? This is the same heavy chain, but by adding a different light chain, we've added diversity. It's another step in diversity. Does that make sense, Elise? Are we all on the same page? Does that make sense? Mm-hmm.
That makes sense, but I have another question. So,
Okay.
let's say when we were pairing light chains for the first time, we went all the way to the second lambda,
Mm-hmm.
but it works. Then we're going up to see if it finds too strong to self. Let's say that one does. Is it just dead because it already went to the second lambda? Or
If it's gone to the second lambda, if all 4 options have been tried and none of them have been as successful, or some of them have been successful but have bound too strongly, it will die. Does that make sense?
Yes.
Are we good? Mary Gates, questions? If you guys have questions, honestly, you listen. Or is it?
Well, Ulysses Clark, I also. have
Yes,
a question.
ask it.
So you make one heavy chain.
And you
just
test that,
copy it to
and you
the-
test that as a B chain, as a pre-B cell. You test the heavy chain, and once that's functional, we know the heavy chain works. With the pseudo light chain. Because the pseudo light chain will always work. We know that it's functional enough to always if we can recombine a B cell receptor or a heavy chain correctly, we can add that pseudo light chain, and if it can send a signal, it's positively selected to move on. Does that make sense?
Yeah.
And then once we have migrated, once we've done that, the RAG recombinase will go in and start trying to make a light chain. The first one it attempts is one of the chromosomes on the kappa. Why? It just is. That's what RAG does. Maybe it's the nearest chromosome to where RAG is made. I don't know. So then it's going to check that chromosome. It's going to rearrange a kappa chain. We're going to put it up there. It doesn't send a signal. So we know that heavy chain was functional. It's a problem with the light chain. So we're going to throw that one away. RAG is going to be signaled to rearrange the other kappa chain. It rearranges the other kappa chain. We put that up there. We test it. Still doesn't send a signal. We throw that away. RAG is told to go to the lambda region. It goes to the lambda region, rearranges that light chain, puts that up there. It sends a signal, but it's too strong. We don't want that. We're going to throw that away. We're going to rearrange the other lambda. We're going to put that up there. It sends a signal, and it's just right. We're going to allow that B cell to migrate on to become, this is now an immature B cell when it has a functional heavy chain and a functional light chain. It is an immature B cell. Okay?
So like I think the question was like, so if that light chain then works, then is it copied over to the other side?
Exactly, because every B cell, every antibody, again, is 2 identical copies of one chain. So we make 2 identical heavy chains, and then we put a pseudo light chain here, a pseudo light chain here, 2 identical. We only make one light chain, we only make one heavy chain for B cell. That gives us two identical antigen binding sites because they're identical, they're mirror images of each other. So when we make the functional light chain, again, notice I didn't draw it on the other side. I only drew it on one side. It would be identical. If I could take that, copy it, and flip it around, it would give us a functional antibody. We have an antigen binding site for the heavy and light chain here, and this allows antibodies to do all the functions that antibodies do. We need 2 antigen binding sites, or 10, or 4, depending on the class of antibodies that we're using, to help do what these antibodies need to do. But every single one has at least 2 identical antigen binding sites. So how many antigens can one antibody, how many unique different antigens can one antibody bind to? 1. Every antibody binds to one unique shape, your mother's face, and only your mother's face. You're not gonna marry Gates, you're not gonna marry Grace's mother. You cannot recognize her, okay? Okay? And the reverse is true. Okay, because you have unique antigen binding sites that allow you, with just a different light chain, to recognize your 2 different mothers. Okay? Does that make sense? And every antibody can bind your mother's face over here and your mother's face over here. Only your mother's face. We're going to learn that I'm lying to you just a little bit in the future. Okay? This step in time, I know it's worse. So we build on this, right? We just build on this. So now we have 3 functional B cells, 3 functional immature B cells that have a light chain that sends a signal but not too strong. Okay? Are we good? What makes it into a mature B cell?
Exposure to its antigen?
Nope. Exposure to its antigen? No. It's not. No. Go ahead, Emma.
If it makes IgD.
If it makes IgD. Now, I know. And Alexia's like, what? Okay, so now, let's look at this. Remember we talked about this. We talked about that we had a VD and J section here on the heavy chain. Okay, this is the heavy chain, this is the mRNA. And to make the mu chain, this is mu. What's next? It would be delta. What's next? Gamma. What's next? And what's last? Okay, so in order to make an IgM heavy chain, we make a transcript of this VDJ section, transcript, intron, transcript of mu. To make an IgD molecule, we use the same VDJ region, only we're going to make this VDJ. Oh, where did that come from? What? Okay, I don't know how that happened. Okay, so this, let me see if I can take this off for right now. We're going to try this. So all we do is transcribe a different constant region. We use the same VDJ region for the heavy chain. We use the different constant region. We simply transcribe and translate a different constant region. Here we're going to use mu to have IgM. Here we're going to use the same VDJ because we know that it works. So all we do is throw that on a different constant region, and now we have IgD. Okay, and when we make IgD, oh gosh, I don't know how to go back. Come on. Okay, we're going to have a different, and now we're going to have IgD with the exact same light chain. Does light chain ever change? We have V, it doesn't have to change. The constant chain will always be, it's a small constant chain. We'll talk about that in a minute. But the variable region is going to be the same. So here we have, I'm going to try and make this into just one. So here we have the variable region on the end of the delta, the same as that is on the IgM. That's the variable region for the heavy chain. And now we're going to make this light chain exactly the same light chain as we made before. We're going to snap that light chain on, and now we have an IgM molecule and an IgD molecule that binds to the same antigen. The only thing we did was transcriptional changes. Transcriptional, instead of making the mu, we're making the delta. And once we have a fully functional mu and delta, and we know that the delta is going to be functional because the mu was functional. The antigen binding site is identical on this delta as it is on the M. So we know that all we have to do is throw a delta heavy chain up there. We now have a IgM, IgD, all naive, mature, and this kind of, we don't know what IgD does. The only thing that we know is IgD is only found on mature naive T cell or B cells. Once this B cell has upregulated an IgD molecule, it is now considered mature naive. And it will migrate, it will be drawn via what chemokine? CR5 to the B cell follicles and wait to become activated. We good? Just go.
So all mature B cells have IgD, okay, but will they all have IgM or is that part?
It has to be, no, it has to express co-dual. The IgM remains expressed, and there's hundreds of copies of this one IgM receptor. There's hundreds of copies of this one IgD receptor. If I went in and looked at your B cells, pulled out some B cells from Aline, I'm going to pull out some B cells from you, and I'm going to look at B cells, and any B cell that has IgM and IgD on it is a mature naive B cell that has never been activated. By definition, it is mature naive by having IgM and IgD. Once we activate it, it becomes differentiated, and we'll talk about that in chapter 6 and 7, or 7 and 8. Does that make sense? If it has IgM and IgD on it, it's mature naive. It's being drawn to the B cell follicles by CXCR5 and waits there to become activated.
Is there something that triggers the development of the IgD? Like, or does it just happen in the first?
It happens in the maturation process. And we don't know if it's a function of IgD to trigger it to leave and that it's now complete. We don't know why IgD is made. We can go in and genetically make mice that express no IgD constant region, and the B cells are what? Completely normal. We don't know why. Well, just completely normal. We don't know what IgD does, but we know it has to do something because over the course of evolution, this wouldn't have remained if it didn't do something. So we think it might have something to do with maturation signals. Mary?
So in the above steps, we made IgM receptors, and then somewhere during the maturation process, another IgD receptor is.
So at this stage, they're immature when they just have the IgM. Heavy and light chain. For IgM to be IgM, it has to have a heavy and a light chain. Okay? If it just has a pseudo light chain, then it's just the heavy chain, the mu heavy chain. Okay? And the heavy chain has a variable and a constant region. All we do is take the same variable region that we created for the mu heavy chain, snap it onto a delta heavy chain, and we have an IgD antibody when we throw the same, because we only make one light chain in every B cell. Once we've made that light chain, that is the only light chain that ever gets made. And so we're going to snap it onto a mu constant region or to a delta constant region, and it's going to have the same antigen binding recognition site. Does that make sense? Yes.
So I have in my notes that after you have the mature B cell with IgM and IgD, and it's activated, you start producing other.
Well, that's activation. We're not going to talk about activation right now. That's you're talking about class switching, and that's following activation of the B cell. Class switching only occurs when the B cell gets activated, and that's chapter 7 and 8. okay? We're not gonna talk about activation right now. All we're talking about is how a B cell gets its spots. How does it get its B cell receptor? It happens in positive and negative selection. In positive and negative selection, we're gonna create a heavy chain. Does it work? Yes. Positively selected. We're gonna put a light chain. Does it work? Yes, no. Yes, positively selected, bind too strongly, negatively selected. Okay, we good? Emma.
So if you rearrange the first capital light chain and it doesn't bind too strongly and it does send a signal, you wouldn't make the other, you wouldn't attempt it again, right? You just move on.
Yes.
Okay.
So what Emma just said was, if I make a successful attempt for a light chain the first time out, everything else is excluded and we move on. And only that light chain, everything else gets hypermethylated and shut down. We'll never make another light chain once we have a functional, usable light chain. Does that make sense? Mm-hmm.
So there's only like 4 options for the light chain.
Yeah, there's only 4 options. You get 4 chances.
So when you say that like the B and J is the recombination of B and J, like that doesn't change. Like I don't know what I mean that.
So, when we're going to make the kappa light chain, in order to make the kappa light chain, let's move again. So let me show you this. Let's look at this. Okay, so now we're looking at this. This is the germline DNA for T cells, and this is the germline DNA for B cells. Okay? When we talk about the chromosome, notice that all the kappa genes are on one chromosome and all the lambda genes are on a different chromosome. We have 2 identical chromosomes from our mom and our dad. We have 2 2s, 2 3s, 2 4s, 2 5s, 2 6s, 2 7s. Okay? In 6 is where our MHC molecules are, and they're co-dominantly expressed. Everything we get is expressed. With these heavy chains, we're going to have a chance, and if you look at the heavy chain of the B cell, notice that we have V sections, we have D sections, and we have J sections. And notice that for the beta chain, we have V sections, D sections, and J sections. Identical, right? But look at, we only have a constant region because the T cell receptor, its job is to recognize antigen. Its job is not to be functional. Whereas these constant regions in a B cell receptor gives functionality to every antibody. It can send an antibody to the mucus, or it can send the antibody to the tissue, or it can send the antibody to a helminth, or it can be involved in ADCC, or it can be involved in, it has, the antibody is the functional component, and it's the constant regions that make that decision of what the final function is. All IgM has the same function. It can bind, it can neutralize, it can activate complement. Other than that, because of that J chain, it can't do much more because the FC region is blocked by the J chain. So it can't opsonize because FC receptors can't bind because the J chain is already there. Does that make sense? We'll talk more about this in just a minute. So let me get this because we have to get through all of this. We have to get through all of this. But when we're talking about, again, for the beta chain, we only have these 2 random constants and they're identical to each other. So once these are made, we have a constant region, we have a variable region. We have a variable beta chain. Notice that the light chain, we have V and J. The alpha chain, we have B and J. So the alpha and the light chain are similar. So what do you think is going to get rearranged first in the T cell pathway? The beta chain. What do you think is going to get made next in the T cell pathway? The alpha chain. Once we test that, and that's when it goes, that's when it becomes, again, we're going to go into a double negative T cell, a double positive T cell, because T cells are different, okay? So, does that make sense? Do we understand now that when we're rearranging this, the first one that's downstream, and again, the way that we draw chromosomes is we say that this is the 5' end, and this is the 3' end, the first constant region that's downstream from this rearranged VDJ exon, and we're going to cut everything else out, is going to be this mu. So that is the constant region that's made first. That's what's tested as a pre-B cell. We're good? For the T cell, we're just going to make a constant region and test this to see if it sends a signal. Did it make a successful rearrangement? Does it send a signal? Yes. No? We throw it away. Okay? So, let's go back and do T cell rearrangement. Okay, so this is the B cell. We're going to do T cell rearrangement, and so we're going to have the same thing. We have a hematopoietic stem cell that's going to become the common lymphoid progenitor. That's going to become a what? It's going to migrate where? And once it's in the thymus, it gets genetic signals to become a T cell, so it becomes a pro. Remember, it's got nothing on it, T cell. It's just sending the signals that you are now a T cell. You cannot be a B cell, you cannot be something else. You are now a T cell. So now it's going to go through the maturation process and it's going to become a pre-T cell by rearranging what? The beta chain. So we're going to make a beta chain rearrangement, VDJ. We're going to have the constant region. And again, they draw all of these immunoglobulin-like domains as like this. That's because when you look at them in a model, it's these loopy things. So this is a blob, this is a blob, this is a blob, this is a blob. So they draw that as a blob on the, as just a circle, right? When we see this expressed as a chain, let's see if we can see the chain. When they draw the chain, see, this blob goes with that. This blob goes with this. Make sense? It's called an immunoglobulin domain. MHC molecules have the same bloopy things. A lot of the immune cells have this bloopy motif. Okay? So when we talk about this, we're going to talk about T cell maturation now. The pre-T cell has the beta chain made. We're going to test that with a pseudo alpha chain. Okay? We'll make a pseudo alpha chain. And again, pseudo alpha chain, just, we know it'll always send a signal. Did that rearrangement of that beta chain work? If it did, then we're going to progress to become a what? An immature T cell. So we're going to have, this is going to become an immature T cell, and we're going to rearrange the alpha chain. Here we have the beta chain. Here, well, let me make the alpha chain. These are now, this is the alpha chain. This is the alpha chain. This is the beta chain that was rearranged. This is the beta chain that was rearranged. This is the beta chain that was rearranged. We now make, but we can do the same thing. We'll clonally expand these, and each alpha chain can be slightly different, depending on the V and the J sections that are chosen. Okay, here, so I'm going to modify these alpha chains so that each one is unique. This one goes this way, this one goes this way, this one goes this way, like that. Each one is different. And by simply making a new alpha chain, we now have combinational diversity. Making a new alpha chain, this is a new T cell receptor. This is a new T cell receptor. This is a new T cell receptor. Okay? And the thing is in this case, if these don't work, then they're just deleted. We make more. Okay? They don't have the kappa and lambda ability to regenerate, to have multiple attempts. Okay? So we have one TCR with one beta chain, one alpha chain. Okay? Now this is, once it has reached that stage, we are going to also upregulate on it two new molecules called what? CD4 and, oops. These are now an immature double positive T cell. This is double negative here because it's expressing neither CD4 or CD8. Okay? So now what we're going to do is we're going to go through positive and negative selection. We're going to do positive selection first. And if this binds to MHC class 1, this is beta 2 microglobulin, what gets downregulated? CD4, because the CD8 molecule is going to recognize a region in the, actually, let me erase that. That's drawn poorly. Okay. We're going to have the CD8 molecule bind to a region on the MHC class 1 that says, I can recognize MHC class 1 of your allele. The T cell receptor here is going to recognize one of your alleles with a self-peptide in it, and all it has to do is send a slight signal to say functional. Again, we just want to know, did we create a T cell receptor? And at this point in time for T cells, the most vital thing is that it recognizes one of your alleles. If it doesn't recognize one of your alleles, T cells are MHC restricted. They can only recognize your alleles. Your T cells are programmed to recognize your HLA alleles. Your T cells will recognize your HLA alleles. If your T cells recognize his alleles, we still delete them in you because all you got is yours. Does that make sense? You only have your alleles. So if they don't recognize your alleles, we can't use them. T cells are not functional unless they recognize MHC. They have to recognize your MHC. If they recognize somebody else's MHC, they're useless to you. Does that make sense? So here we recognize one of these 6 HLA, A, B, and C alleles to make it a class CD8 positive T cytotoxic cell. In the case of this cell, we're going to recognize MHC class 2, which has 2 chains. And what are we going to downregulate when we recognize this T cell receptor recognizes this with a kind of a self-peptide in it? We're going to downregulate the CD8 molecule and we're going to express it as a CD4 molecule, and that becomes a T helper cell. We call it a TH0. It is now mature, naive, single positive. Mature, naive, single positive. Yes, Elise.
To be clear, the CD8, when it's expressed, makes this cell a T cytokine?
A T cytotoxic cell.
Cytotoxic.
Yes.
And then the CD4 makes it a T helper cell.
Yes, these are now programmed because of the molecule that they bind, of the HLA molecule that they bind, are programmed to either be a T cytotoxic cell or a T helper cell based on the MHC molecule, which is why we say they're restricted by their MHC. T helper cells can only recognize MHC because the CD4 molecule binds to a region on the MHC DR, DP, or DQ, and that binding is necessary for activation. If it can't bind to that region, that co-receptor, and the reason we call it a co-receptor, because it has to bind along with the T cell receptor in order to send a signal. If that CD4 molecule, if we eliminate it genetically, the cell can't function. The cell can't become activated. That co-receptor, the T cell receptor complex, the T cell receptor, CD3 and the zeta chains, must signal with the CD4 molecule for a T helper cell to become activated. Aileen? So we have T cell receptor complex is a T cell receptor, but the T cell receptor can't send a signal. What sends a signal? CD3 and the zeta chains with the ITAMs.
So what step are CD3 and the zeta chains like upper?
Really, all of them, so whenever they're being tested, the CD3, that's a great question. You can think that the CD3 and the zeta chains have to be here because we're testing whether or not it can send a signal.
The first like positive stuff?
So as soon as we get a beta chain rearranged, we have to see if it's functional. We put a pseudo light chain or pseudo alpha chain up there. The CD3, we know the CD3 molecule, the signaling machinery works because it always works. It's set, okay? That doesn't have to be tested. We know that works, so we'll throw that on the receptors that we're making, and we're seeing if it can bind to antigen. Does that make sense? Elise?
So the CD3 and the beta chains are always there as long as the heavy chain's working?
As long as the beta chain.
The beta chain. So it's present throughout this entire thing.
Because it has to be, because what we're testing are whether these can send signals. Can they be functional? Can they send a signal into the nucleus that makes this receptor work? That's what we're testing. So we do need T cells and B cells cannot send a signal on their own. The only thing they can do is they can activate their signaling molecules. They're complex. For T cells, that's CD3 and the zeta chains. For B cells, it's Ig alpha and Ig beta. All of those have ITAMs, and we'll talk about the signaling pathway in chapter 5. Okay, yes, Emma?
So is the alpha chain recombination similar to the light chain recombination in a way that, like, if it's you have multiple attempts and if the first
No. So really, we're gonna say for the T cell receptor is it gets run one recombination attempt, and if it doesn't function, we kill it. Okay? For T cell receptor, it's just a allelic exclusion. Whatever gets rearranged, the other one, if it doesn't work, we throw it away. We'll just say for T cells, it's completely different. And I don't know if that's actually true, but I'm going to say that that's true for this class. Okay? Elise?
I'm confused, but I don't know what I'm confused about.
Okay, so let's just walk through. Let's go back through this figure.
So if the CD4 and the CD8 are expressed together, what does that do?
So that's exactly what happens. That's a double positive.
So what is a double positive?
Let me check. I'm going to get to. So here we have the stem cell, hematopoietic stem cell. This is the double negative pro T cell. This is the pre-T cell. What chain is that? The beta. And what's associated with it? Pseudo alpha. And so is CD3 and the zeta chain have to be there? They do, because we're sending a signal into the nucleus that says we have functionally rearranged successfully a beta chain. Then we're going to upregulate. Once we've rearranged the alpha chain, along with the alpha chain rearrangement is the co-expression of both CD4 and CD8. That is the double positive. And that's when, for B cells, real positive selection occurs, because now we have functional receptors, but now both receptors must be tested on MHC. If they can't bind to your MHC alleles in your body, we will throw it away, because the only way that we can activate a T cell is through MHC molecules. If they don't recognize your MHC molecules, they are useless to us. They're useless to you, and we will delete them. Once they recognize an allele, class 1 or class 2, they become differentiated finally into a T cytotoxic cell, CD8 positive T cell, or T helper cell, binds to MHC class 2, CD4 positive. By definition, those are the two things that identify a CD8 is a CD8 binds to MHC class 1, a CD4 binds to MHC class 2. And it's, again, you're learning see, Jane, run. It's actually much more complicated down here, okay? Much more complicated down here. This is the smallest byte. This is the least acceptable unit to understand how a T cell receptor gets made. So, we have VDJ recombination that gives us one step of variability. We have the alpha and the beta chain or we have the heavy and the light chain that gives us chain combination. Now we're going to talk about junctional diversity. So, when we snap the D and the J together, we're going to take a random D and a random J, and in that animation, that's a really good animation from those immuno animations if you haven't seen them yet. Go to the immuno animation, and there's signals on either side of the V, D, J sections that RAG binds. RAG1 binds here, RAG2 binds here. It pulls the genetic information together, and then it chops that chromosome off in between. And each side forms a hairpin loop. And when it forms a hairpin loop, then what happens? So that's what RAG does. So let's go through what happens when this is being created. So here, we're taking just, for simplicity's sake, they took V1, D1, and J1, okay? So we're gonna join D1 and J1. D and J always come together first, okay? So we're gonna join V and J together. And the way that we do this is we are going to have the RAG enzyme is going to pull this genetic material. There's the D that we're going to get, that's D1. This is J1. It's going to take this receptor, it's going to pull it around this way, and it's going to lop off the intervening chromosome, and that becomes, it's called the signal sequence, but every time we see circles of DNA floating inside the cytoplasm of a cell, it tells us that rearrangement's going on, because we've lopped off part of the chromosome, and it just floats away. Okay? Now we have, but we can't have two unopened. Actually, this is a way of adding diversity. So by making a hairpin loop, RAG adds a hairpin loop here. Now you guys understand how DNA has 4 basic nucleotides, A, T, G, and C, right? So what we're going to do is then we're going to have something called TDT. So we've made, these are going to, well, we're going to open this up, and we're going to have the RAG combination is going to cut the DNA on either side of this chromosome and flip that hairpin out to make, we call it a palindrome. And you're like, wait a minute, what do you mean by a palindrome? It means that if we look at this, that's a T, C, G, A, what would go opposite this A? T. What would go opposite this G? C. What would go opposite this? G. So it reads in DNA, it reads the same way forwards and backwards. So this TCGA would have an identical TCGA on the opposite side if we filled it in, right? So RAG is going to make those P sequences. So we're going to have the palindromic sequences made by the RAG to yield that DNA hairpin. It's going to cleave that hairpin to form this palindromic series, which we just added diversity. Because before it was TCCACA, and now the sequence reads what? TCGA. So we already have diversity just by taking that and flipping out and making that palindrome. So Mary Grace up here. Here we have the sequence in the DNA would read TCCACAG. Now what we've cut this DNA at this point, we've eradicated this, and all we have is TC and its nucleotides, its homologous nucleotides on the other side, okay? So we're going to cut this here, and we're going to flip those out, and now we have a palindrome, because in this way, it would have been TCGA, so it reads the same in this direction as it does in this direction. Does that make sense? So now we have a palindrome. We call it a P sequence, because it's a palindrome. And we've already created diversity, because just by lopping that off and flipping this out is now 2 different nucleotides, and we know that 1 base pair change is going to change the amino acids following, right? So diversity created. But we're going to even add more diversity to this. So the next thing that happens, and we did the same thing on the other end. We created a palindrome on the other end. And instead of it reading T-A-C-A-C-A-G, it now reads T-A-T-A. Diversity created. Okay? So, what does Harry Potter say? It'll come to me. So, now what happens? Sorry, it's, what is it called? Yeah. Anyways, it'll come to me. Again, brain part. Now what we're going to do is we're going to have TDT. As long as you remember the enzyme, TDT. Because the actual name is terminal deoxyribonucleotidyltransferase. If you break that down, we're going to go to the terminal deoxyribonucleic acid. We're going to add rando DNA nucleotides. Random. Blah, 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 blah. We don't know. It's random. Whatever happens, happens. Okay? So, we're going to add random DNA. The TDT just adds random bases. Okay? And then what happens is now that we've added TDT by these nucleotides, we're going to have, trying to get binding. DNA always wants to be double-stranded. And it just binds by hydrogen bonding. But we always know G binds to what? C. And we always know that T binds to? So we're going to find regions in here where even a couple of nucleotide sequence randomly come together someplace in this newly created region. It could be over here. It could be over here. It could be over wherever we find homologous combination, binding. So we're going to create this random binding, and it's just that these two are where it binds. We're going to cut off. We're going to have exonucleotides. Remove the ones that don't bind correctly, because DNA has to be perfect. C's must bind to G's, and G's must bind to C's. T's must bind to A's, and vice versa. So then, now that we've created this region of homology, we're going to have DNA polymerase come in, fill in the gaps. And we have created junctional diversity. And because this is random, this is where I'm saying so many millions of different possibilities can occur. Because anytime we add one extra base, depending on the base, we can change the amino acid downstream completely. So this has just happened between this has just happened. So we have the D section here. We have the J section here. We have the P nucleotides on either side. We have the N, we call them N nucleotides because N stands for any, and it's N to the X. However many random nucleotides has been put in there to create this new junction that will only exist between this one particular junction in one particular B cell or one particular T cell. We're going to do the same thing once we have this J with junctional diversity, P, I just call it PNP. We're going to do the same thing between this and the V. And now we have an exon that is your variable region. Does that make sense? Are we good, Elise? Anna? So again, we have 3 levels. All B cell receptors and all T cell receptors have 3 levels of diversity. They have the random, whichever V1, D1, J1 was selected, or V2, J1, D1 selected, or V45, J32, J6, D whatever. So you're going to pick randomly. Okay? Karen?
So this is all showing how diversity is added to
This is how junctional diversity is
Okay,
created.
is junctional diversity the same thing as diversity in the CDR3 region?
Yes. So this is one of the things the book talks about, is one of the regions that's hyper variable, and it makes sense that this is hyper variable because it's random. That these are what cause the CDR, the hyper variability in those loops that make every antibody slightly different from every other antibody. along with the V's. If we just have the V's, the D's, and the J section, eventually we only have, if you look at the book, it tells you how many combinations that could be. It's very low. By adding this junctional diversity, we just log levels higher of diversity. Does that make sense? Okay, so let's look at this. If we look at this, the book talks about this. So if we just have, again, once we add removal of nucleotides, once we have these addition of p nucleotides, we can get a TCR up to, we can get 10 to the 16th. And let's do 10 to the 16th. 10 to the 16th is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, right? I don't even know how many that is. I call it a buttload. Okay? Because of that extra diversity, because of these, ability to make these, if we just had this, we'd only have 3 to the 6th, or 6 to the, again, to the 6th. We need that junctional diversity to create the Brazilians of B cell receptors and T cell receptors, because you need a B cell that will recognize any epitope that you ever run into. Ever. In your whole life. Courtney, are you good? Do you have questions? No? You know?
Is combinatorial diversity different from the diversity created by like which heavy chains with which light chain or alpha?
So you can think, again, it's hard, so let's name these for us. So in the book, they have different names, but let's name them so we remember everything. I like to think of VDJ as combinational diversity, because you can take this V with this D with this J, which you combine. So here we're doing V1, D1, J1. Here we're doing, the other one was V36D something else. Okay, so that's that. And then we have like additional diversity where we add a heavy chain with a light chain. So chain diversity, or which chain associates with it, you can think of it as whatever we want. It doesn't, sometimes not very clear in the way they describe it in the book. So we can think of it as change. Whatever light chain goes with whatever heavy chain. And then while we're creating those light chains and those heavy chains, we're going to add additional junctional diversity, because at every junction between the J and the D and the V, we're going to create junctional diversity with the P and P nucleotides. Does that make sense? Mm-hmm.
So in total, we would have 3 different types of diversity that would create the antibodies.
Yes, and T cell receptors. The same thing is happening for T cell receptors. Okay? So again, here we have, because of these extra, we have 50 numbers of J's on the alpha chain that increases the level of diversity of combinational diversity, simply because there's so many extra 50 of the J's. So here we have fewer of the light, but what's going to happen is B cells have an additional way of creating diversity. So B cells can go in, and once we've created this, we're done. There is no change in the T cell receptor. This is it for T cells. This is all we get. If we can't make the T cells that will recognize a pathogen, then we're screwed. Because this is all we have, these 3 mechanisms of diversity. Because antibodies are so important, we actually have another level of diversity that we're going to add once we get activation of B cells. Okay? So there's another level of B cell diversity that happens only in B cells. It's called affinity maturation. Okay? So we have the combinational VDJ, we have the chains, we have the junctional diversity, we have RAG making the palindrome P, cutting the DNA, making the hairpin, flipping out that, making palindromes. We have TDT adding the N nucleotides. We have homologous recombination. The 2 sides are going to find homology. We have exonucleases chew out the bad ones that aren't bound. We have DNA polymerase fill in that, creates the junction. Pretty straightforward. In the book, it explains all of this in words. For me, I'm not good with words. I need pictures of how that happens. So that's why I included that other figure from someplace else. Are we good? Okay. So we have a T cell receptor. And again, if you look at the B cell, we're going to go back over. We have the stem cell, the pro B cell, because it's now been told it's B cell. It's going to start rearranging its heavy chains on the chromosome for B cells. Here we're going to activate RAG1 and 2, create VDJ rearrangement. Junctional diversity is occurring while that VD and J is occurring at the same time. So combinational diversity and junctional diversity occurs at the same time, right? So we are creating our heavy chain. We're testing it with a pseudo light chain to see if it sends a signal. If it sends a signal, it's positively selected to mature. We make a whole bunch of them, we start throwing light chains up. Every different light chain is a different chain combination. We have a functional heavy chain. By just making different light chains, we have now created a whole universe of B cells that all have the same heavy chain, but have different light chains, therefore they're different. So we now have an immature B cell with a rearranged kappa 1 or kappa 2 or lambda 1 or lambda 2, 1. Once it's functional, it's set and the rest of them don't go forward. Okay? So we have 2 kappas and then the lambda. And then once we make that, we have an immature B cell that just has IgM on the surface. By transcribing the B, D, and J that was created for the light chain, for the heavy chain, and the VJ that was created, the light chain that was created here and tested here, the same identical light chain will be put on the delta. The heavy chain will now be the delta chain with the same VD and J section as the IgM has. We throw up the same identical light chain, and now we have a mature B cell, a mature naive B cell, because it has simply two different heavy chains with the same VD and J region on them, identical light chains. How many antigens, how many unique antigens can this B cell bind to? 1. Always 1. Always 1. It doesn't matter that the heavy chains are different, the antigen binding site is the same, and they're the same in the one arm of the antibody and the other arm of the antibody, and we call that the variable region, because that's where we get variation. We call this the constant region because because we know that there's only 5 classes: mu, delta, gamma, epsilon, and alpha. They're constant. Your alpha looks exactly like your alpha, that looks exactly like your alpha. Now the weird thing is, and this is what we talk about with monoclonal antibodies, when we talk about monoclonal antibodies, mice have mu, delta, gamma, epsilon, and alpha, but their chains are glycosylated differently and look slightly different from our chains. You can think of them as hairy with tails. So they look like us, only they're hairy with tails. So it would just be like if Savannah was hairy and had a tail. She would look just like Savannah, but she's hairy and has a tail. Is that different from us? Would we notice that? The immune system notices that as well. So antibodies made in mice trigger, that doesn't look like that human antibody, that looks like something freaky weird. Sorry. Okay. The same thing is true in goat. Sorry, you're the goat now, the greatest of all time. Okay. Goat antibodies would have horns and a body, which look different than human. You can't put goat antibodies into human because we notice those as different. The constant regions look different. The variable regions are always going to be different because depending on what this person has seen, what this animal has seen, you guys will all have different variable regions depending on, Mary Gage, you might have been exposed to the mumps and you may never have been exposed to the mumps, Haley. And so your immune response will make variable regions that bind to epitopes on the mumps And Haley will never have made those. The same thing is true with tetanus or vaccines, or when we talk about vaccines. This is how it all works. Alyssa, are we good? Are we good? Yes.
So, what part of this whole process would create, so I understand that the variable region is what recognizes an antigen, but what makes each receptor different from each one? Like what part of this?
The variable region. So let's go back to the antibodies at the beginning. Let's go back to this. So when we look at the T cell receptor, this is the variable beta. So VDJ recombination has gone in on that aspect of the T cell receptor. So this is VDJ recombined. What is over here? This is the variable alpha, so it would be V and J. And that creates the variable region that recognizes the MHC molecule and its peptide. Okay? So it looks like that is just set, but you can think of this now as being this shape. That's the actual shape of that because of VDJ and VJ recombination. They're just drawing it in the immunoglobulin-like domains because it's lumpy like that. But in this region, you can see these loops up here. These loops are where junctional diversity has occurred.
That junctional diversity allows each T cell to recognize a different antigen.
The VDJ recombination and the junctional diversity
Together.
and the combination of chains allow this T cell receptor to recognize an allele of your MHC that it recognized in positive selection and eventually a pathogenic peptide. Does that make sense?
So all of those factors together.
All of those factors together. We're good?
Yeah.
Does everybody get that? So, VDJ, VJ, junctional diversity, this beta chain with this light chain all come together to create a unique T cell receptor that will be the only T cell receptor that this T cell will ever be able to make. And it will be a unique snowflake, just like you guys are all unique snowflakes. I can tell all of you guys apart, even though you all have one beta chain and one alpha chain, because the variable regions on your face allow me to recognize small, subtle changes in the topography that creates something that I recognize. Does that make sense? Okay, so this is the T cell receptor. These two constant regions never change because the T cell receptor is only supposed to be a receptor. Its job is not to go out and do something. Its job is to recognize MHC and peptide. Okay? The other thing that happens is one of the things that happens in positive and negative selection. When we talk about positive and negative selection for T cell receptors, when it goes through the thymus, it's going to go through the thymus, and up in the thymus, there's going to be a region. You just need to know that in positive selection, it's just going to recognize MHC class 1 and 2. So we're going to have certain thymic cells that express MHC class 2 we call them thymocytes. Immature T cells are thymocytes to test their double negatives on. Can I bind to MHC? We have to have MHC class 2 in there to make CD4 cells, right? So we're going to have MHC class 1 and MHC class 2 up in this region of the thymus, okay? And then the other thing that's going to happen is in negative selection, what gets interesting is that MHC 1 and 2 start expressing self-peptides from all over the body, not just the thymus. We're going to talk about a transcription factor that allows DNA and proteins to be made for the liver proteins, muscle proteins, nervous proteins, all the proteins that your T cells are going to MHC and what is self, and if they bind to self too strongly here, they're negatively selective. Here, they're just recognizing MHC. Can they recognize your MHC alleles? Your particular alleles. You have HLA A1, B2, C3. You have HLA 4672, B from your mom, right? So you get one set. Do your T cells recognize that set of MHC alleles, A, B, and C from your mom, A, B, and C from your dad? They will recognize one. They only have to recognize one, because that shape is what they recognize. Every allele is different and unique. It's going to recognize one and a random, and again, a very small self-peptide. It's a pseudo, you can think of it as a pseudopeptide, like a pseudo-alpha chain and a pseudo-light chain. We're going to put a pseudopeptide in there because an MHC molecule has to have a peptide in it to be displayed, right? So we're going to put just a random common peptide that'll fit in any MHC molecule and allow it to be displayed. And then once we do that, then all of your MHC molecules will start displaying your MHC muscle peptides, your MHC liver peptides that have your anchor residues that your T cells will recognize, and if they bind too strongly, we will delete, because we cannot have self-reactive T cells, because remember, T cells are what regulate the immune response. If we have self-reactive T cells out there, then we are going to have autoreactive immune response, and we're going to have disease. We're going to have problems. Does that make sense? Okay, so this is, we're going to talk about what that transcription factor is later on in the chapter, but this is where negative selection occurs, and this is where we make sure we don't react to self. Okay, does that make sense? It's going to be the T cells that regulate whether B cells can bind to self a little bit. They have to bind to self a little bit to send a signal because they're being tested in the bone marrow, right? But without a T cell to say you can progress on, B cells can't become fully activated. So this restriction against self is going to regulate whether or not B cells can go on and make antibodies against self. Does that make sense? We'll get to that more again. This is the first pass through. We're going to come back to it again and again and again as we go through the class. Okay? Okay, so this is B cell, this is the T cell receptor, and notice this T cell receptor is recognizing this part of this particular allele of MHC class 1. What kind of T cell is this? It's a T cytotoxic cell, and so it would have a CD8 molecule that comes and binds. There's a region on this constant region of the alpha chain that binds the CD8 molecule that sends co-receptor, receptor. CD3 is going to be on either side of that T cell receptor. The zeta chain is going to be in there too. So we have the CD3 T cell complex along with the co-receptor. We're going to talk about co-stimulatory molecules after this exam. Are we good? Unfortunately, things, antigen, antibodies sound similar but are different. Co-receptor, co-stimulatory molecules sound similar but are different. It's easy to remember the co-receptor is associated with the receptor, the T cell receptor. Okay? Does that make sense? Good. Okay, so let's go back. We're going to walk through this chapter. Okay, we're going to talk about this is the variable region. To this hinge region is the variable fragment. Because when we were trying to identify what immunoglobulins were, we first found out they were made during an immune response and they were proteins, so we called them immunoglobulins. So we then started digesting them with different enzymes. And we found if we use one specific enzyme, this one, again, these are disulfide bonds. These disulfide bonds kept this region together, and this was the variable region, and we called it the fragment of it also of antigen binding, or the FAB region, because it's variable and it also bound to antigen. And then when we looked at this region, what do we call that region? The constant region. So that's the fragment that's constant. Now, is there a constant region for the heavy chain in the FAB? There is. This constant region, the CH1 constant region, is just there to help the variable region be displayed. And it's really analogous to the constant region on the T cell receptor. Does that make sense? Because again, that's all we need for antigen binding. We need a constant region and a variable region. Technically, if we stuck this into a transmembrane region, this would be a T cell receptor, right? If it was a T cell, if it was beta chain in. Does that make sense? Whoops. So there is a constant region, and when this class switches, that entire constant region will be changed to, if this is a mu, because it says it's a mu. Here we can switch this to a gamma. Oh, this is still heavy. We can switch this to a gamma. Notice that there's still going to be a constant region, constant region, constant region. But there's a different, we simply put a different heavy chain onto the same variable heavy region that was created. This variable region is identical to this variable region. The only thing that's changed is the heavy constant has changed from an IgM to IgG. This is now an IgG because this is now a gamma chain. Does that make sense? We still have the variable region, the FAB region. We still have the same variable region that we created during positive and negative selection. We don't want that to change because that's what was tested in positive and negative selection. Did we make a functional variable region? This is what creates the antigen binding site, your mama space. Okay? So we've created your mama space. We still want to make an antibody that binds to your mama space. We just need to get it into the blood and tissues. That's where IgG goes. So when we talk about IgG, so we have the FC region, we have the FAB region, we have the variable, heavy, variable light, constant light, and this light chain will be the only light chain ever created for every B cell. It makes one light chain, it throws one light chain up. When class switching occurs, same light chain. We can switch the heavy chain and keep the light chain because we need that for our antigen binding to understand the rest of your mama space. Okay? So we have light chains, we have different antibodies. Once we start, and again, when we talk about these bumps like this, it's just this blobby thing. So here we have two identical blobby things. Technically, they're different colors. They should be the same color because this is a copy of this. They're identical to each other. This light chain, notice, is the same green color. They're only drawing it in different colors to let you know that there's two of them. This is identical to this. That light chain is identical to that light chain. Those antigen binding sites are identical to each other on one antibody. Good? Okay, so we've got that. We've got antibodies. So we're going to say in this class, IgA is a dimer. Okay? IgA is a dimer and it's supposed to be in the mucus. IgD is just a naive, mature BCR. If there's IgD present, by definition, it is a naive, mature B cell. IgE is defense against helmets. IgG is the highest concentration, and it's supposed to be right in blood and tissues. You do not have to know the different isoforms. This is the highest total antibody in entire body. Because we throw, even though it's not in the serum, because IgA's job is not to be in the serum. IgA's job is to get into the secretions. We put IgA in your vaginal secretions. We put IgA in your seminal fluid. We put IgA in your respiratory tract. We put microgram amounts of IgA into your gut every day. It's in every secretion, and if I dried you down and separated all the antibodies, the highest total would be IgA by a large margin. IgG is sleek. It's a monomer. It's only a monomer. Its job is to, when we get inflammation, is to sneak into the tissues and start binding and activating complement. IgG can do everything. IgG does everything against everything. It is the antibody of choice if we need ADCC, if we need opsonization. It's the only antibody that opsonizes. It's the only antibody, it's one of two antibodies that can activate complement, IgG and IgM. Okay? It can activate complement. It can agglutinate, it can neutralize. IgM can do that. Okay? And we're going to also talk in later, the rest of this stuff is going to come in later in chapter 7 and 8. But notice that IgM is always a pentamer. And again, this light chain is the same as this light chain is the same as this light chain is the same as this light chain. Multiple copies, 10 copies of one light chain. 10 copies of one heavy chain, 10 antigen binding sites, 5 antibodies joined with the J chain. For dimer, for this, it says mainly a dimer. It says it can also be a monomer and trimer. Ignore that. It's mainly, we always look for dimers. Generally, it's always a dimer. And when we see antibodies, this is what we see. We see, this is what it looks like. It has a J chain. And when we look at it under electron microscopy, that's what it looks like. This is an antigen binding site. I can tell you that this is the FAB region. 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 This is one heavy and light chain. This is one heavy and light chain. This is the J chain right there. Exactly like that. When we look at IgM, this is the 10. This is the J chain. We have an antibody here, an antibody here, an antibody here, an antibody here. 1, 2, 3, 4. Technically, I think I drew an extra antibody. Okay, there's only 5. There's a J chain, and we snap 5 of them on. It's the same J chain. It's just that for mu, we're going to snap 5 on, and for alpha, we're going to snap 2 on.
So every antibody has 2 FAB or just?
Every antibody has at least 2 antigen binding sites. Every antibody has one FAB region. That FAB region contains 2 antigen binding sites. Okay, does that make sense? Every antibody has one FC region. Okay, are we good? Okay, and then these are affinity. The reason that we talk about the KD, the more negative it is, the better it binds. So we have, this is the affinity, is how well it binds. IgG goes through affinity maturation, so it binds really strongly. IgM is the first antibody always made, and it doesn't go through affinity maturation. That's why we need avidity, where we have 10 weak antigen binding sites, but the combination of 10 allows us to bind fairly strongly to something, and these are all the same epitope, antigen. By definition, this one antibody can only bind to one unique epitope But there'll be thousands of epitopes on the surface of a bacterium. There'll be thousands of epitopes on the surface of a virus. Anna?
So even in this, there's only one FC region?
So in this, there's one FC region that's IgM. There's one FAB region, but there's 5 copies of that FAB region. Every FAB region has 2 antigen binding sites. So there's 10 antigen binding sites. All 10 of these antigen binding sites are binding to the same mama's face, to the same epitope. Only one epitope, but 10 copies of that epitope. Are we good? Mary?
So each.
So again, remember, every antibody makes one heavy chain and one light chain. Identical antigen binding sites.
Yes.
We have 5 identical antibodies snapped onto a J chain.
Okay.
So 5 would be 10 antigen binding sites, 10 identical. So because these are all exactly like each other, they're all identical to each other, they have to bind the same shape.
So one pentamer will bind,
10 copies.
so 10 binding sites will bind to one microbe, and that's high avidity.
It's high, so it's high avidity because
Okay.
with 10 of them binding, it enhances the binding strength. Does that make sense? Yes.
So affinity is just like
How well the antigen binding site
that
binds.
a single antigen binding site binds. Avidity is the strength of
Multiple.
Is that just specific for like I guess like one antibody with polymer, or is it?
So they actually, in immunology, we talk about affinity and avidity all the time of every antibody. How well it binds and affinity as well.
So like, could you talk about, when you're talking about avidity, is it only like, okay, the 2 antigen binding sites are like IgA, or is it like I could have like 300 IgAs binding to the same?
You could have 300 IgAs binding to the same.
But could you
Or in-
refer
or
to
multiple.
those like 300, could you refer to the avidity of those 300 as a group, or would you have to just?
Okay, we're progressing into math, and as soon as we start into math, I draw the line. All you need to know is the higher the negative number, 11, 12, minus 13, higher affinity, stronger binding.
Okay.
The lower the number, minus 8, minus 6, the weaker the affinity, the more need for avidity.
Okay.
Does that make sense?
Yeah.
Which is why we have probably 10. And it really is mainly about IgM. Because IgA will also go through affinity maturation. It will become stronger as well. Okay? Okay, so the last thing that we're going to do here is we're going to talk about monoclonals. When we talk about monoclonals, we can create a B cell antibody response specific for whatever thing we want to. And we normally do this in mice, and we'll do this in mice, and if we want to identify how many CD4 positive T cells you have, I'll get an antibody that is specific for the CD4 molecule. I'll label it with a red fluorescent protein on the FC region. When it binds to all of my CD4, it will tell me how many total CD4 cells I have in a population, and we use a laser to differentiate them, or a microscope, okay? However, the problem with producing them so B cells have a normal, activated B cell has a normal half-life, and it will die. We fuse these B cells that have been activated by an immune response. We have effector B cells making plasma cells. We will fuse these to a lymphoma, and again, so it's a melanoma, so it's a mutant, myeloma, it's a mutant B cell that is no longer producing B antibodies, but it's immortal. It will grow and divide and grow and divide and grow and divide as long as it's in certain media. It has lost the ability to make one enzyme, and we simply add it to the media to grow these myelomas. So as long as we're growing them with the presence of that enzyme, they can grow and divide immortally. Thousands, we can make buttloads. We can actually make it so you can see these myelomas in a test tube. However, these B cells, the plasma cells will die, the myelomas will live forever, but they're missing an enzyme. We fuse them together and we make this hybridoma. We take away the enzyme in the media. So if it's not fused, the myelomas will die because they don't have the ability to make that enzyme that they need. The B cells will die because they die naturally after a certain amount of time. Only the fused cells will grow on to grow and divide and grow and divide and secrete the antibodies that we want. And then we put every individual hybridoma into a well. We let them clonally expand and make antibody, and then we take a little sip out of each well and we see what those antibodies bind to. We identify that the specificity of this antibody is for this protein, and this is the protein that we want to identify. So we're going to just keep these and throw those away because those might have been did something different that we didn't want. Okay? And then we have this plate and we simply have our now fused cells, myeloma, B cell, producing antibodies, make our specific antibodies. We can't use these in humans because they are furry with a tail. They are mouse myelomas. So what we've done, and when you went in and looked at those, and we call all monoclonal antibodies, every organism, every therapy that we use these monoclonal antibodies for, we're going to have a monoclonal antibody, but we can humanize these monoclonal antibodies by taking out the mouse furry tail, random, mu, delta, gamma, epsilon, alpha, whoops, and we can put in only, do we want it to be IgG human? Guess what we're going to put in? Just a human IgG constant region. That way it has to class switch to IgG. Does that make sense? Okay. I think that is it. These all, if you look these up, these all, in order to be used in humans, have to be humanized monoclonal antibodies. And these are extremely, extremely expensive. It can cost hundreds of thousands of dollars. It can be $1,000, $20,000 a month in treatments with these monoclonal antibodies. Okay? Have fun at the soccer game. Text me if something happens. I will be on alert for the next 36 hours. Don't say anything about anybody else's soccer team, though. They take it very seriously here. Okay?
And I was like, oh, that sounds like cool. And I brought it up to me and I found out we weren't going to see it. And I was like, well, I'm like, buying nice Korea, Nigeria, New Jersey.